today. But let's move on, guys. We need to see the future. So today we will start a new course, and this course will be led by José Onuchic and also by Paul Whitford. So I'm going to introduce them, and they are going to split the course. One day will be one, the other day will be the other one, but they'll be here together, okay? So José Nelson is a professor, has a background both in physics and electrical engineer with a bachelor degree from University of São Paulo. He's Brazilian. And he got his master's degree in applied physics from USP as well, and then a PhD in chemistry from Caltech, the California Institute of Technology in the USA. Currently, he is the Harry and Olga Rice Professor in Physics at Rice University in Texas in the United States. He's also the co-director of the Center of Theoretical Biological Physics. So that's why he's here, right? He works in many biological scales, from molecular level interactions to cellular systems, to organized multicellular structures, focus on questions related to protein folding and function mechanisms. He also works on the theory of chemical reactions and condensed matter with emphasis on biological electron transfer. Okay, now I'm going to introduce Paul Whitford. Paul, it's also my pleasure to introduce Paul Whitford. Professor Paul is a physicist by training with a bachelor from, oh my God, that's really hard to say, the university where you got your...
Worcester Polytech.
Worcester Polytech Institute, and a PhD in physics from the University of California. Currently, he's associate professor at the Northeastern University in the United States, and also a member of the Center for Theoretical Biological Physics at Rice University. In his work, Paul researches the energetic properties of biomolecular dynamics through a combination of theoretical modeling and high-performance computing. That's why he's here as well. So help me to welcome Professor Paul and Professor José Nelson.
So let's do the following idea here with the students here. I'll do English because it's easier for them, but everybody here can stop me anytime, and you're allowed to ask questions in Portuguese. So if you feel more comfortable, just go for it. And Paul is not here, but he needs to get his training in Portuguese better, so you can ask him questions in Portuguese also. He's back there to make sure he gets good training, okay? So let's ask a question to everybody here. How much do you know about proteins? Do you know anything? Do you know nothing? What? No. Let me ask a very simple question. Should I start the lecture assuming you know nothing, or should I start the lecture assuming you know what proteins are? So let's do a light flight over it. I'm going to go these first slides very fast, but if you don't know anything, just stop me. And this is just to refresh your memory, because Paul will be talking about models, and it's very important to understand what we are modeling. Right? So basically, when we start with biology, you have all the different scales. Okay, and probably on this course, you have people commenting all possible different scales here. And the goal we have in the center is how can we model all the way across all these scales, okay? And We are going to stay on this course, mostly on protein, but try to come to ribosomes or things like that that tend to be a little bit on that scale. The lecture today will be an overview of the ideas. And then for a couple of days, starting tomorrow or a little bit after tomorrow, Paul is going to really start to tell you how to actually simulate these systems, how to go to high power computing and stuff like that. I think it's going to be something very useful for you guys to do. So if you think about proteins, you know that, but I'm going to try to refresh your memory because that's very important for things. Proteins are, people like to say that proteins are a heteropolymer of amino acids. Okay, where an amino acid is this particular unit where you have an amino group, a carboxyl group, they match at the end, they lose the water and they make a chain. And what makes these different beads is whatever is hanging from the side. Okay, so it's a necklace of beads of different colors. And this different necklace of beads of different colors, they're going to have interactions. Now, that's what a biochemistry book tells you. But the reality is that that's basically, for proteins, actually, they have to fold into a unique state. And that's what makes them differently. Okay? So let me tell you a little bit more as we move along. These amino acids have different colors. They have different types. And in biology, there are 20 types they have to do it. I'm not going to go into details. But the most important thing is that they tend to have groups that are hydrophobic. Right? It's like oil coming together. They have groups that are polar. They can be polarized. They are hydrophilic. And then they have charged groups that can be positive or negative. Okay? And the interesting question is, do you need all these different groups for folding? Or actually, you need 20 groups just because you're going to do some function. You need for chemistry. Okay? Several people that are working on amino acids discover they can actually fold proteins into a particular shape, any shape you have, just using 5 kinds of amino acids. So what they showed is that basically, you need to have hydrophobic groups, and you need to have the polar groups, that basic charge or... And people learn that in order to have full packing and design, you need at least 2 kinds of hydrophobic groups, the ones that have a small side chain and the ones that have a large side chain in order to do packing. And on the polar groups, they need at least 1 positively charged and 1 negatively charged. And then they figure out they need glycine. Glycine is the amino acid that has the side group just a hydrogen, so it's the most flexible one. So in order to fold the protein, you need that flexibility at some point. So this tells you that basically you can actually fold proteins. You don't need all these amino acids. You need just to do something to bring these things to come together. So it's not a surprise that the simple models of protein folding, people try to read amino acids, they say they are polar or non-polar, right? Hydrophobic or hydrophilic. And we're going to go into that. Now, how many degrees of freedom do I need as I model these things at very simple models, right? Basically, I have to deal with these things. So the first thing people notice, and I'm going to go a little bit faster here, is that basically these amino acids can get themselves organized in such a way that you try to do periodic orientations of them. They can form hydrogen bonds, right, between the main backbone. And if you want to do periodic change, there are only 2 forms you have, the alpha helices, where you go IL+4, or beta strands. Doesn't mean they cannot have any other forms, but means periodic one. If you want something that repeats itself, these are the possibilities. So let's keep that in mind. The second part I want to say is a very interesting thing. Because this nitrogen here, you see when you make the bond here, has a partially aromatic notice. It's sort of, you have an interaction on this group between this and this nitrogen and this carbon. They form these things in a resonance, and these things forms a plane. Okay, so if you have a look here, you start to observe, you have one amino acid, here starts another one, the alpha carbon is in the middle, here's the bond connect then. But these things has a planar nature. That means the only degrees of freedom you have per amino acid is this angle phi and this angle psi. Okay? So that tells you that when you're orienting these things to first approximation, you can think about that as 2 degrees of freedom. Right? And you're moving these groups around as you start to get these things packed. Right? So it tells you, although you have many, many degrees of freedom, if you're not worried about the size of each bond, you assume the covalent bonds are all conserved, these are the things you have to do it. And there are lots of people that try to do simulations just in terms of phi psi angles, but these are actually very complicated calculations, because as you have angles, you know that basically you have all these problems on doing simulations with angles. So this is just a number you can find in any book that tell you the typical distance they have, and you start to observe that when you describe these amino acids, here are 4 of them, you have sort of the angles between them, and these angles tend to be conserved, and these phi psi angles are dihedral angles. That means they're the angles that things move. You create a plane, right? Every 4 groups, 3 of them make a plane. Then you figure out what's the angle the other one is moving out of the plane, right? That's the dihedral angles that you're computing when you're doing these simulations. So I don't want to go into many details of that, but I just want people to keep in mind these things as we move along. Now, here is, just for curiosity, and I think this is something that people try to have a look, to figure out, if you look at proteins, how much amino acids exists of certain kinds. And when you observe, just out of curiosity, there are some amino acids, like leucine, that have to happen often because they tend to be very important for structure in terms of charge. But then the big amino acids, like, for example, tryptophan or thyroid, they have these big aromatic groups. You only have them when they need, because they're hard to pack, so they show up less often, okay? And I just want to remind you. And then you have the cysteines that happens very rarely, because they're very useful groups for chemistry, but they are very reactive. You don't want to have them unless you need, unless you want to make those sulfur bridges into proteins. So there are lots of tricks that the proteins use as they design themselves. Okay? So just for your curiosity, they're all the structures that you have. If you think about a protein, they can have a chain. That's what you call the linear version of a protein. It just gives you a list of amino acids. These things can get organized as what we call secondary structures. They are local structures. Okay? These local structures can pack around each other. That's what we call the tertiary structure. And then multiple proteins can come together and form combination of multiple proteins, and that's what we call the quaternary structure. Okay? And the beta structures, they tend to be parallel structures, and you can form things where you come one way and turn around. These are anti-parallel, or they can go around a big way and come parallel to these trains. And these are the structures that you have as you move around proteins. So this is just for your curiosity. We don't want to overdo, but we want to come. And I'll be glad to email all you guys these slides, so don't worry about it. I'll make PDF all of them and email, so don't worry about having these things. You can get that in any book, but this is just a summary. Okay, and you have these topologies as we have. If you have quaternary structure, there are many things like proteins can form a monomer, but they can domain swap. They can get another monomer, and instead of binding with you, it can bind with another partner protein the same way, right? Because the interface is the same. So there are many structures. They're very tricky as they do, and helps with stability as they move around. Okay? There are also some proteins, you're not going to talk too much about it, but most of the proteins I told you about it, they tend to be in solution. Means they want the hydrophilic groups outside, the hydrophobic inside. Some of them have to be around membranes, so they have to play around with this hydrophobicity in order to be stable into membranes. They form fibrous structures. Proteins are the machines. They are the workers, okay? Think about your DNA keeps the information, tell you what to do, but the work is done by proteins. right? All the work inside the body is done by proteins. Now people learn the sugars are also involved and some fats, but Now, one thing we're going to be talking about it on the simulations, and we'll be talking about lots of protein simulations, is how they're able to fold and self-assemble. And there's lots of questions about predicting structures of proteins going around. I will have some time to tell a little bit more about it. Maybe on the last lecture, I'll tell you about all the new tools and how things have done, how you have doing data analysis, how we develop sequence information to predict structures, how these ideas were developed about 18 years ago, now is used by AlphaFold to do big predictions, what they call using covariance. So there are lots of things that base these physical ideas are now used together with artificial intelligence, machine learning, to do structures. And here is just to finish this story, to remember that proteins do a lot of function. Okay? So this is a very short introduction that we want to put in this case of what proteins are. Okay? So now let's try to say about it, I don't care about proteins. I don't care about molecular biology. I care about physics. Actually, I care about both. Okay? So let's sort of, oops, what happens here? Something got unhappy. Let's try to do something about it. Zoom. Let's do the following thing. Stop, share. Okay. We were able to solve a Zoom crisis. Okay. So this is what I told you. Now, let's try to build models. So when you start to build models, these are the slides I just showed you, you come into experimental models. So the first thing you observe, let's do some ideas of phase transition. So let's everybody think about phase transitions. Okay, so these are some experiments to show you what in the laboratory people learned about proteins. So what they learn about proteins is they start to observe there is some free energy for these proteins to fold. And people are interested in what this transition is. So what they tell you here is this free energy. There's the free energy of the native state versus the free energy of when the protein is unfolded. And you change that with temperature is equal RT or KT, makes no difference what just the units you're doing, over K. Over K is the equilibrium constant, is how much you have of unfolded versus folded. Okay? So when you have K equals 1, you expect to have a system where half is folded and half is unfolded. Now, don't worry, this one, just look this one in the top here, just tells you as you change the temperature and you are looking at the plot of the fraction, right? The case is the ratio of the fractions, tells you how much you have denature and how much you have in nature. And you observe you have the sigmoid plot, where at high temperature you're fully unfolded, fully denatured, and low temperature you're fully stable into the structure, and you have a transition that looks like a sigmoid. Okay? And you claim the sharper this transition is, the more cooperative you are. Okay. I have a bunch of physicists here. Who is going to tell me the difference between a first-order phase transition and a second-order phase transition? Who knows what the difference is? Tell me one first-order phase transition that you know all the time.
Okay, so the difference is, well, in statistical physics, at least equilibrium statistical physics, we have a partition function which describes the distribution of states in the system, the probability distribution of states. And we say that a transition is nth order when the transition, basically, when the partition function becomes discontinuous at the nth derivative. So it's a kind of...
He gave you a mathematical definition. Let's work with the mathematical definition. Let's give you a practical definition. What's the most common first order transition we deal all the time? Ice, water. A first order phase transition has latent heat. Okay? That means at one particular temperature, the system has energy being transferred in terms of water, in terms of order. So when you have water going to ice, at one point, if I want to melt ice into water, I have to add heat to the system to create entropy to do it. So there's one temperature that you just do that transition. You have latent heat. A second order phase transition you don't have. It's a smooth thing. You just keep, like magnetization. You just smoothly start to add heat. You also change the temperature, you're getting more and more ordered, but you go in a continuous way. And what he said is a first order phase transition, because you have latent heat, has a discontinuity on the first derivative of the partition function, and that's why you call a first order phase transition. The second one is continuous. You only have a discontinuity on the second derivative of the partition function. So mathematically, as we do the problems with partition functions, that's what's going to happen to you, okay? So keep that in mind. That's the mathematics of it. But the most important thing is discontinuity comes because of latent heat. So, in proteins, that's a plot of a protein unfolding. And here, you measure the heat capacity, and you see you have a peak here, and this area is the amount of heat that's given to the protein at that point. Now, a protein is a finite system. It's not an infinite system like water. So this change of transition is not a very sharp transition like in water. 0 degrees Celsius, you go from ice to the other one, you have a little bit of a peak, an area to do it, but it's a first order like. You go into a transition where you need heat to transfer into this way. That's why you call first order like. You're not a real phase transition, but you have this peak that comes here. Right? And this latent heat that comes basically in an exchange of enthalpy versus entropy, right? Because you expect at that point the free energies are the same. Right? So you're exchanging between these two terms as you go through that transition. So proteins are first order like because they have this peak here at this point. There is this heat transfer almost at a constant temperature that tells you what people call order disorder. What's the consequence of that? The consequence of that is that when you look at protein getting ordered, it's either, at least in equilibrium solutions, you're fully disordered or you're fully ordered. You don't have a protein going through 10% order, 20% order, 30% order. That's what a first order like transition looks like. It's the fact that you go between a disordered state and an ordered state, I can describe as a two-state system. Okay, so now I'm getting a problem that was totally biological at that time, and I'm telling you in a very happy way that this protein now is first order like. Now, let's continue. And our friend here told you about a partition function. And Paul is going to tell you lots about sampling and computing states and finding order and disorder. So why don't we try to define basic physics here? Okay, let's try to describe some basic statistical physics to help our case in a very simple way to try to move to that. Okay? Here's one page of all you have of formal definitions of what I just told you. I'm not going to go into details, but that's for you to remember that basically You can have CP or you can have CV. There's a system constant pressure, constant temperature, makes no difference here. The important thing is that you have this latent heat and the terms get connected to that, okay? Let's not go on these details too much. Let's try to make our life simpler. This is, you're going to find any physical chemistry book or physics book as they move around. But I actually want to make your life simpler for what we're going to do. So always remember what I have. The important thing I want to remember from here is not constant pressure, constant volume. That I think I can let you Paul go over the details of that. But I'll tell you that when you have that transition, you have a exchange between order and enthalpy. Okay? That means in a system, the tendency is disorder, right? It's just like your bedroom. If you know nothing about it, in half a day, everything's in the middle, right? Unless you put some energy to get everything folded and put it on the drawers, the tendency of disorder is enormous. That means that when a protein folds, that first order transition tells you you are losing disorder, you are losing entropy, and you have to pay that price with energy. That's what I want you to keep in mind. All these other things are here. It's good for you to learn if you get into details, but you're not going to have enough time to do a lot of thermodynamics here just for the fun of it. But you got the basic idea. We go through this order transition, then now we have to formalize, and we have to compute it. And we have to be able to compute those numbers, and that's a lot of stuff we're going to learn this week, how we actually compute these things. How do I find what's my density of states? How many states do I have? How do I do these things? Okay? Now, I can make your life very complicated, right? That's basic. I'm very good about that. But I'm not going to try not to do it. So here is an interaction. You are having these things. Like I told you, I don't care if you do constant volume or constant pressure. For our practical purposes, they are the same for what we're doing here. We actually can do simulations both ways. And you have this energy comes through interactions when the chain comes together. Now, I'm going to do a simplification here. I could start with a system where I treat my full protein and water system and I compute everything. In my first part, I'm going to simplify. I'm going to get my effective system as being just a protein. And it means that you're going to have interactions, like hydrophobic interactions. They're actually effective interactions. I'm just going to talk about order and disorder of the protein. And the entropy of the solvent, I'm going to put on these effective interactions. When you have a hydrophobic interaction, when two non-polar groups come together, if you put water, oil and oil coming together, they're not coming together because they like each other. It's because the water is trying to get together with each other, and they're trying to meet. But effectively, you can treat it as a potential in the proteins when hydrophobic groups come together as one interaction. So we're going to treat as interactions as energy on a lot of descriptions we'll be doing here. Okay? So let's try to think about it. I'm going to jump these slides because I don't want. This tells you that you have you can work these interactions. These are hydrophobic groups with hydrophilic groups. These are experiments that people tell you if you put one group in oil and put in water, which ones are happy to be in water, and there are lots of experiments on that. I can go spend an hour doing that, but I actually want to do physics with you guys today, so I'm going to jump over to tell you there are these interactions. There's a very interesting thing if you look at membranes, proteins. You can find if you have hydrophobic groups or hydrophilic groups based on the table you had before, and it's not a surprise that everything that goes through the membrane is hydrophobic and everything goes in solution is hydrophilic. So that scale tells you on average. You can see it's not a constant interaction. That's what I want to show you. We are going to treat as constant interaction the first simple models, but tells you that you have, as you have the situations, as you put in solution, you try to use that. So the simplest idea that I told you is not sufficient is to tell you that hydrophobic groups try to get together and hydrophobic groups try to get or in solution or with each other. Okay. Now, are we happy? That's a very simple description of protein interactions they come along. I can make them more complicated. I can add the details. I can add details that basically, in addition to this hydrophobic interaction, you need packing. So if things fit together, there's an extra factors that come in there. But the simpler idea, to have this idea in our mind, you're going to do complicated simulations with Pod where you put all these other details, but let's keep the same idea. You have to have interaction to help you to get order in order to beat that disorder. Okay? Is that something we are keeping in mind? Is that clear? So you're going open books if you will say, hydrogen bonds stabilize native structure of the protein, and if our interactions compete into the tight packing, things coming together. Hydrogen bonds actually is something that fools you. People love to say, I told you about hydrophobic packing. How about hydrogen bonds? Well, let me tell you one thing that's very important to remember. Why you have all these hydrogen bonds? When the proteins unfold, that CO group and that NH group, that amino group, they're very happy in solution. The CO group has all the waters that can make hydrogen bonds. They're extremely happy campers in solution. However, the hydrophobic group say, we want to get together. Right? So the hydrophobic group, their side chains, try to pack together. And that CO and NH that are in solution say, heck, now I'm unhappy. I had all these hydrogen bonds that make me happy. And now these guys bring me to the core. I'm losing all that water. What do I do? So a lot of the hydrogen bonds that come on beta strands and alpha helices and other things, they're sort of minimizing losses. They're trying to compensate for the hydrogen bonds they have in solution to do that. So that's a very interesting thing. A hydrogen bond gives you about anything from 3 to 5 kilocalories per bond. Okay, let's get people here. Units, the energy is here. The energy is here. Basic unit that we need in order, RT or KBT. How much is a KBT? How much is a KBT? Yes, room temperature.
Like 1/40.
Okay, 1/40 electron volts. Okay? 0.6 kilocalories per mole. You'll be looking at things from chemistry. Okay? Good numbers to remember. Good numbers to remember. Okay? We are paying prices on those units. Okay? So when I tell you that typical hydrogen bonds can give you from 3 to 5 kilocalories per mole, I'm telling you that you're giving something from 5 to 8 kTs. So you know how much you have. However, that's a hydrogen bond in vacuum. When you're in solution, you get just a fraction of kilocalories per mole. You get less than a kT stabilization because you have to take what you lost from solution to make it to the center. Okay? So these are numbers to think about it. But still, as this protein organize, bring the hydrophobic groups to try to stabilize itself, try also to take care of the hydrogen bonds, otherwise a big price that you pay because you're breaking these hydrogen bonds with solution. Right? And you have to take that into account. So you're seeing, I'm throwing a bunch of concepts for you. I don't expect you to understand all the details of them, but keep that in mind. You can learn that as you come along. As a theoretician, or as anybody doing a simulation, you have to know what are the facts that are helping you. You have packing. You have hydrophobic groups coming together. That's what I call vulnerable interactions. Things have to pack together. Why is that? Why packing is an important thing? If you do any simulation or any work, you can do that with a liquid of hard spheres. If you create an empty pocket vacuum, That's entropically very expensive. Right? So you have three interactions you're trying to do. You're trying to satisfy the hydrogen bonds. You're trying to get these hydrophobic groups together, and you're trying to fill up the space as much as you can, because creating pockets is a very unhappy thing for your life, right? So these are numbers they're going to learn as you do simulations, okay? The models are going to do, and I'm going to include all the effects deployed, but that's something for you to keep on the back of your mind as we move and do models and we do simulations on that. Now, I can do statistical mechanics as complicated as you want it. But this slide, actually, a lot of this slide I used the first time in a course of statistical physics. So I was very fortunate. When I was a very young student in graduate school, in a course of using path integrals for quantum mechanics and statistical physics. When I was at Caltech, that was 1982, as on the last class that Richard Feynman taught about path integrals. And he had a slide very similar to that, so that's all you have to know. Now, calculate these things can get very complicated. But in practice, that's all you have to know. So what it tells you is basically, you're going to have a partition function that Feynman always hated the word Z and he used Q. Don't ask me why. It's just because I say it's just choose the letter you want. That partition function is a very interesting object. It's a complete abstract object I'm going to describe to you. So on the canonical ensemble, I could describe any different ensembles. We are going to live on the canonical ensemble. There's a system put on a thermal bath at constant temperature, right? I don't know what they want. I'll ignore it. So that term here has a very interesting thing. This first term here, what I call W of EJ, that's what you get from the microcanonical ensemble. That comes from the following idea. I get a system, okay, that's very important for simulation. And I try to enumerate all possible states. If I have a discrete system, it's very nice, right? And I will tell you about the discrete system a little bit. If I have a discrete system, I can actually count. Right? If I have a bunch of balls, black and yellows, and I say how many ways can I order it, that's a discrete system. You can make all possible combinations, you can count. And then, as I count that states, I can categorize those states based on whatever quantity you want. If I'm going to deal on a canonical ensemble, I have at least to categorize that states based on what energy they have. Now, that tells me I'm going to count. That W is a counting of how many states has a certain energy EJ. Okay? So I'm describing here, I have several possible values of this energy. 1 to S, and I'm going to say how many states has a given energy. Paul is going to give you lectures for you tomorrow. He can say, oh, that's not enough. To understand this problem, I also have to count how this W depends on EJ and some other quantities, like one I'm going to call Q, that tells you how ordered my protein is. And understand, now you can write your system as a function of as many variables you want. But as you want that thing, like Matthew Fisher, one of the most famous statistical physicists in the world, said, statistical physics is just counting. Can be very complicated counting, but it's just counting. And I love the ideas in theoretical physics to develop how you do that counting, okay? Now, in the canonical ensemble, you're going to tell you that the energy is something that defines order. Okay? So that tells you you have a bias into the system. Energy is a bias into the system. Tells you that states at low energy tend to be more likely to be occupied than states at high energy. But this occupation depends on that factor E2 minus beta EJ. That's E2 minus EJ over KBT, or RT. Makes no difference, the unit that you're using here. So basically, you have to measure how much that energy, if you are room temperatures, in units of 0.6 kilocalories per mole. Right? So that's what gives your system. So as you are seeing here into these systems here, the system will tell you, you can see, if this temperature goes to infinity, that term becomes 1. Every state has equal probability. That means your system is going to tell you. But another interesting thing it's telling you here is that on the canonical ensemble, states at the same energy have the equal likelihood to be occupied. That's why the canonical ensemble is the simplest case of a maximum entropy case. What you have is, other than the constraint is the energy here, the leftover is maximum disorder. Now, I can do lots of problems doing maximum entropy. I may tell you something if I have time this week. But when people come tell you about maximum entropy, that's what they mean is the following. You have some constraints that come. On the case of the canonical ensemble is the energy, but then everything else is maximum disorder. It means if the energy is the same, then every state is equally like. Okay? And now, if you compute that term, you can find Pj, the probability to have a certain energy J. That's going to be that term divided by the full sum, the Q. It's just normalization. You can find the value of any quantity, average of these Pj's, and you can find the entropy of the states using the quantity minus K P log P. So in a sense, and I can go in much more details, this is what we'll be computing. Like I said, this W may have other variables as we come along, as we put together, right? Because you may want to see not only how the energy changes, but how other things go. But the constraint is just the energy. The bias into the system that tells you which states are occupied is based on energy. That's the canonical ensemble. Okay, and what we hope into proteins, there's a temperature. What you see is, if I put my temperature equals 0, that term goes to 1. If I put my temperature equals infinity, if I put that, because that term goes to 0, if I put the temperature equals to 0, only the low energy state is going to survive, because that's going to be the smallest number. So it's when do I go by having the low energy state occupied versus having all the states unoccupied. And when I talk about order disorder, is that you go from these states where you have many states occupied to this one where very few states occupied in one single transition. That's a first order transition that I hope to see on my system, right? That's what the experiments I showed in the beginning. That's what latent heat means. You're switching in a very short transition in temperature from one very ordered state to one very disordered state. Okay? So let's keep that into mind as we move along. Now, Paul is going to tell you about big simulations, but I will tell you about simpler model you can think about just for you to create a feeling for what we are doing. So the simplest models we had in proteins were trying to get polymers on 2 dimensions, square lattice, or 3-dimensional cubic lattice, and see how they organize themselves on this very simple lattice. They're not real proteins, just to understand the order and disorder on polymeric systems. So here's just some case. Let me give an example. Let's do a problem just for you to understand the ideas of what we are computing, okay? Just to get ourselves into shape to start tomorrow with real big simulations. Let's understand what we'll be computing. Because these big simulations are just following the same physical concepts that come here. So I'm going to find the simplest model that I can think in terms of order. Let's imagine that I have a chain. It's a protein. So, like I told you, it's a necklace of beads of different colors, okay? Now, if I have to be a heteropolymer, the minimum I need is at least 2 different kinds of beads, right? If I have one kind of beads, I'm homopolar. And I'm going to tell here red and green beads. Or blue. Actually, I don't think it's looking more blue than green, but I think it's just the PowerPoint fight itself. Okay, what I'm telling you is I can tell you the red, one is hydrophobic, one is polar, so I can imagine a potential, a very simple potential, where it tells you beads of the same color try together with beads of same colors, and beads of different colors repel each other. Okay, so that's the simplest potential just to think about, like hydrophobic, hydrophilic. But that's just for us to do a mental, theoretical exercise to understand what we'll be doing when we come to real systems, okay? So, now I can come to you and say, you have a big computer, so try to do a very complicated problem. That's what you do on every simulation you have on your system, continue or discrete. Here I have a bead that's 6 long, a chain that's 6 long, okay? And I just chose these colors here. And I'm going to do on this potential, I'm going to tell on this potential a very simple equation, okay? I'm going to tell on this potential, then red beads, when they come together and form contacts in space, red is attractive. That's like, say, hydrophobics like to come together. And every other interaction, red, blue, and blue, blue, is 0. That's what people call the HP potential. So you tell hydrophilic groups come together. The polar groups are either happy, they stay with another polar group, or with water makes no difference. Right? That's a very simple potential. So what I'm doing here? So now I ask you to solve this problem. Okay? So in statistical physics now, you're going to solve this problem. And the first thing you need, in order to compute this WE, number of states, you have to find all the states. Okay? Now, on this problem here, where I just have a chain of 6 beads, you can do that very easily. You can go around and find states that are symmetrically different, and you can enumerate all those states. So what this problem is telling you here, and that's what we're going to do in every simulation, you're going to enumerate all possible states you have. If you have a discrete state, you're going to have to find a resolution, you're going to try to beam based on some quantity, but the concept is the same. Right? So basically, so what I figure out is if you work your way around, you're going to find these systems has 36 possible states that you can actually put this chain, start from the one that's fully extended to the one that's fully packed, and you put all those states here together. So the first thing you do is counting. Is creating all these states and figuring out how many states you have. That's the art of looking order, disorder, phase transition to proteins. Even when you have folding or functional states and they're moving around, you're going to have this density of states. Okay? So that's in front of you, and you have it. Now, I need to write a partition function for that state. Remember, I wrote you I have to write a partition function. So to write a partition function, I want to categorize that states based on what? Based on their energy. So what you see is, on this particular problem, that I just put you as an example, for this particular chain, you end up and see that 28 of those states, you don't have any red group coming together with a red group. Right? So all those states are going to have an energy equals to 0 based on the unit I put you here. Okay? All these states are going to have energy. They are going to form a contact, epsilon 0. Okay? They are going to become more attractive. Okay? Now, I copied this from a book, and that's just to annoy your life. I love to annoy your life to understand that reference can be epsilon 0 is a positive number or a negative number. It's an attractive number, so it's a negative number, right? So you see, if you read here, looks like this is a number smaller than 1. Actually, this number is a number larger than 1, right? You're telling you that those states are starting to get more and more occupied, right? It's an awful notation, but I love to put notations to confuse you here, and then you think about it always in time. So what you start to observe is, as you start to see on that state, if the temperature goes to infinity, all these numbers become 1. If the temperature goes to 0, this one goes to infinity faster than everybody else, only that state is going to survive. So that's your partition function. Right? That's exactly the term I did you before. Right? And tells you that basically, if you are very high temperature, the term with the largest number of states start to dominate. At very low temperature, the states start to. So this is a number where you can actually compute exactly. Right? It's a very simple problem, okay? I can do and sit on my own, right? And even old people like me that don't like to do large simulations, joking, can do it. I can do on the privacy of your room, you can enumerate the states. Now, why I like that slide? I like that slide that tells you for lots of systems, that's what we're trying to compute. And to compute these states is an art. You have to make sure you sample enough. You have to make sure you have these numbers correct. It's very complicated sometimes, but that's what you're after. And if I have this partition function now, as soon as I compute my partition function, I can compute everything. Every thermodynamic parameter, every transition. Then my problem is solved. My life, out of this number, I'm in business. So a lot of the stuff, when you do it, is either compute Q or the probabilities or something proportional to Q, the probabilities like I showed before. Right? But these are the terms that you're trying to have and try to compute in order to do your life. So now, what do I mean by that? That means that now I can compute. Remember what I told you, delta G is minus RT, the probability of being native versus being denative. So basically, if I go back one state, it's going to come native, it's going to come from the chi-square over Q, and this is just that term, right? But it's just to remember, what I'm computing here is just this term. For the full native, for the full disorder, but you can compute it for the 3 states. So the system tells me now, I can compute these probabilities. It's straightforward, right? Now I can get delta G fold. That's native versus no native. It's going to come from the probability of being native, probability of being completely native. That's the one that's 0, and the one with 2 contacts. And when you put this term there, you put KT and you cancel, that's what you get. Okay? And you can find your transition temperature exactly when this term is equal to 0. Right? That's what you have eco-population to do. Now, it's very interesting because now on this problem, you can see that this is a very simple problem, very few states. So the first order transition is not that perfect. You see the heat capacity is still very broad, but give you the idea of a first order transition, right? This problem is give you an idea. And you see that basically, from the top there, you go from unfolded to folded as you go, right? And you see, as the temperature comes down, you get ordered. You see at the transition, you populate a little bit of your intermediate state. But you also see that the folded state goes up, the unfolded goes down. You see a first order transition come out of this very simple model. Okay? So on this problem here, for you, you have a feeling of a lot of the stuff we are going to do on this course. A lot of the quantities you'll be computing on much more complex problems than that. But these are the quantities you're going to be looking for. Right? And if you have a partition function, you can come and compute that quantity. And that's not necessarily very easy in some problems. That can be very complicated. On this case here, we can enumerate. So let's stop here. Questions? Any questions? Do you want to go back? Any questions? Yes?
So just to be sure, the counting of these states, I guess you're counting a state as different from another as long as there is, I guess, you put like a chain, a new element perpendicular to the last.
Is in solution.
Yeah.
Okay? So I'm going not count states basically, so you should be able to rotate them or do anything like that. They are the same. You cannot differentiate that. Okay? So they are symmetrically distinct states that happen there. If I had a medium that is organized, then you have to count different. But here we just count them as being a one state. Okay?
Okay, so you're assuming just an isotropic
Exactly.
medium, and then you can count off
Exactly.
these orientational.
Exactly.
Okay.
And basically, and the interesting part is that I'm not going to do in lattice, but you could actually do simulation where you can actually count how many times you occupy the states as long as you have a ergodic sampling of the medium. But here you can actually count them, okay? Any other question? So, yes, please.
How can we possibly count these for proteins, like thousands of amino acids and thousands of interactions?
You know, I'm not smart enough to answer this question. I'm going to let you Paul tell you that yesterday. So clearly, you're going to have some sampling techniques that allowed you to do that. Okay? And then the sampling techniques become a problem. There are difficulties on doing that. It's going to address a lot of these things when you run sampling, when you run simulations, when you do these things. So for example, I can tell you that when you do big simulations, for example, sometimes when you do sampling, you have to do sampling at lower temperatures, you have to do sampling higher temperatures, because doing low temperature, you never visit the high energy states. Then you're doing high temperature, but you don't visit the low energy states. Then you have to match the simulations to actually build the full density of states. So there are lots of tricks that come into do that. So what you're asking is exactly what will be addressed in lots of the simulation techniques. But I won't tell you on this lecture what we are going to try to compute. I don't want some say, I'm going to compute the density of states today. Why I'm computing the density of states? So here I hope that you get the concept why we need those quantities. Anything you do, any thermodynamic property, you have to have the partition function tells you how often the states are occupied, how they're doing, how you're competing between one or the other, how do you have an order transition. But I love here, because on this plot here and the plot before, you understand what a phase transition is, what that means, because see how your probabilities go. If you have a system that's much larger, much more comparative, the heat capacity could be much straighter, much more localized, right? It's going to be, but the concept is the same. Okay? That's what we have as a challenge ahead of us. Okay? You see, it's a competition between order and disorder. But I can tell you it's order, disorder, but at least here, I hope that if you just look the last couple of slides, you understand that order, disorder really has a quantity to do it. And there's a temperature for that transition. That's when the two free energies, order and disorder, are equally likely, right? If you think about it, order and disorder comes, the transition temperature is when these two states are equal. Right? At low temperature, you're dominated by that state. At high temperature, you're dominated by that state. At the phase transition, they are equal. That's exactly when you're switching from one to the other. Right? And the other thing I was trying to tell you from the next slide is that these intermediate states that you're partially ordered are never too populate. In most of the cases, you're either folded or unfolded. But yes, but there is a time, there is some intermediate state here in the middle. Yes, there is, because it's a finite system, right? But the concept is, and right, you're dominated by one, or you're dominated by the other. You go through this full order and disorder transition. This is a finite system. Ours is very finite, right? I have a chain with 6 beads with just one very simple interaction. I cannot get a full, very sharp transition, but it's enough for you to get a feeling of what you have. You're actually moving between unfolded and folded, and you're never populating too much the intermediate state. It's really a full transition between order and disorder. That's where we're going to see a lot into protein folding, a lot into many other properties, a lot into effects of allosteric effects and other things as you go through this transition of order and disorder. And now you're going to have many other things with partial order, partial disorder. All these things go along, but that's the simplest idea in there. So this is an assignment I used to give in a course of mine, but basically, you can write this, it's simple. You can see that basically if you want to make your life miserable, you can change beads and try to do it, see how the counting goes. But in general, I don't want you guys to spend too much time on that, so that's a good place to stop here. Let me do a stop share here. So if I understand, I have, what, 10 more minutes? 10 more minutes. What? 20? Okay, good. Even better. So this first part is actually a regular course, would be 3 or 4 lectures, right? But we have a week to cover a lot of stuff. But I hope that you guys got a feeling for it. Okay, you're going to have lots of problems, and that applies for a lot of problems in statistical physics, not only proteins, everything. These are the quantities you're trying to compute as you go to the system of phase transition, order, disorder, how you get it, how these quantities come to you, how they come to you. And I love this problem that's based ridiculous 6 beads Hamiltonian, but you can see all the concepts in there. So now, let's start. We're trying to do So now,
José?
yes.
A question that was from the one of the first slides. You mentioned that somehow it was proven or it was shown that you needed only 5 kind of amino acids to build it up. Could you expand on how that was shown? If that was shown computationally or if it was?
Okay, let me tell you a story. When this field started, now there are lots of people doing protein folding, but when it started, there were very, very small groups involved on that. And one of the guys that was doing the first big experiments in protein folding is actually well known these days, is Dave Baker. He has what they call the Rosetta protein design software. A lot of people use it. It's a very famous software. So he was very interested on doing that. So in those days, he got a very small protein they call SH3. It's a very small beta strand proteins. And this protein has 3 parts. And he started to say, can I build things that fold into that shape with very few amino acids? Now people do this much easily. So he started replacing these things. And he figured out he could only stabilize that particular structure just with 5 groups of amino acids. And then he discovered that basically he needed at least 5. Otherwise, the system couldn't get stable. And it makes sense because you need charges, and you have to be able to balance charge. So you need to have a positive and a negative. The hydrophobic need at least 2 to pack. That was an interesting number. There was a question how many you need to actually do minimal packing, and need glycine. So he actually did experimentally these things. And he asked a question I'm going to ask you guys, since you asked that question. He created many, many different sequences, just with 5 amino acids, that were able to fold the same way as the native protein. Now I'm going to give to you guys and use all your intuition. These proteins, they were designed. They were more stable or less stable than the native protein. What's your guess? You create a bunch of proteins that fold exactly the same shape artificially. Now, these proteins that you're creating, let's say I create 20 of them, and you measure their stability, right? It's like I'll measure their TM, the temperature where they're stable, right? The more stable they are, the higher is TM, right? You have to go to higher temperature to break them, right? Do you think these designed proteins are more stable or less stable than the wild type, than the native protein? The answer is you had some more stable and some less stable. That makes sense? Let's think how evolution works. Remember, your proteins are selected by evolution. What evolution tries to do? What's the goal of evolution? Is to be functional with the least amount of effort. So it means that proteins in real life, they have to have enough stability. After they have enough stability, there's no need to keep looking for better ones. So there's always some people with this idea in evolution always looks for the best. No, evolution is just like us, right? Basically, I do as much effort as I need and then I stop. Right? I could do a little bit better on my homework, but I think I can get away with that. Right? If I stay on the lab 80 hours a week, it's better, but if I can survive with 35, I can be on the bar the other. So, right? So in a sense, it's a very interesting result. Right? Because evolution has to find a good solution to survive. The goal is to survive. And sometimes, to have something optimal is not the best for survival. The best for survival is something that works and is robust. Right? So we can go for 2 hours lectures or discussion what do we mean that robust is. I will not do that now, because every time when people say something is robust, it has to be something robust to whatever. Whatever is the stresses that you are living under, right? Right? You have a protein that lives on the limit of stability, that I need a precise medium to survive. If I get drunk this evening, I will die. Because, right, so I have to be able to be stable under different conditions, right? That's what I mean robust. So there's a lot of different things that come into your survival. Okay. So now, with that introduction, and on my last 10 minutes, I can start to move on that. Okay? We'll let Paul tell about real proteins tomorrow a little bit more, but we can start to move on that, just to start to get a feeling for these things. So the first thing I try to tell you in a first order transition is the difference between a random heteropolymer. So the beauty of protein folding, when the physicists start to talk about it, a lot of times on structural prediction, people tell the following thing. If I give you a protein, a sequence, a one-dimensional sequence of protein, can you tell me what the three-dimensional structure is? Right? That's a structural prediction problem. However, that's an easy problem. In many cases, if I ask you that question, I'm telling you that that particular sequence of amino acids has a unique structure. So when we came from physics on the old days, right, basically, I got on the 90s when I became assistant professor and started to move on that. We switched the question around. Let's do a tougher question, becomes much more interesting in terms of physics. Give you a sequence. How do I know if that sequence is a protein or not? How do I know if that sequence is able to evolve into a unique structure? So I'm trying to contrast here by saying, what's the difference between a random heteropolymer and a protein? So what you have here, the random hetero problem is a problem of frustration. Okay? So you guys remember when you're in kindergarten, you have a bunch of kids. At that age, my wife taught kindergarten all her life. At that age, you understand that girls get with the girls, the boys get with the boys. Changes happen afterwards, doesn't make a difference. So the question is, if you get a bunch of people, and they have some of their friends and some of their enemies, and you tell them all to give hands, and you say, get close to a friend. So what happens here is, if I get something where I say blue like blue and green like green, every blue tries to get it with a blue, every green tries to get it with a green. However, you're giving hands. You have a chain. And you're going to end up have some green next to blue because you cannot satisfy all the interactions. So when in physics we say frustrate interactions, you actually end up with a frustrate bit. You are next to someone, you don't want to be next to it. So the word frustrate in physics, frustration, is actually a word that has a real meaning if you think the social part of this problem. So in a random heteropolymer, if I get this chain and I say, start the clock, try to get together with a friend, you're going to see that every blue tries to get it with a blue, every green tries to get it with a green. That's exactly what I told you before, hydrophobic, hydrophilic. If I put water and oil, they try to separate and minimize the surface between them. However, because of the chain effects, you're going to end up with some contacts that are unhappy, like the one here on the top. If I redo this experiment many, many times, whatever is the state you get may not be the same, because where the frustrated interactions happen may be different. So that means that all these low energy states are states that are structurally very different, because where the bad interactions happen are different. So in a frustrated system, the low energy states, the ones that try to minimize the global energy, they are not able to locally minimize all the energies. Every minimal in global energy you try to find is going to be in some local areas, it's going to be unhappy. So unfrustrated system, as you minimize the global energy, you're also able to minimize the local ones. So what happens is you have a bunch of states here, they're very different. Now, this problem becomes a big problem. First of all, if I ask you to find which are these low energy states of all those states, is a computationally nightmare. Right? And I can tell you, if you have a system that has absolutely no bias towards any states, you actually have to look at every state. Right? So it's a total nightmare. But not only is it very hard to find, it's irrelevant. Because if I change the conditions of my system a little bit, change the temperature a little bit, or make more polar, change the amount of acidity of the system, change the pH a little bit, this state may go up and this one may come down. So I spend all the computer time in the world to find the low energy state, and I find a very non-robust answer. So not only is a computationally extremely demanding problem is a worthless exercise. Okay? So real proteins try to do something else. Real proteins try to find one state that's stable relative to everything else. And that's what we call, these are sequences that are able to minimize frustrations. And these are sequences that tells you that local states are also trying to minimize. So it means you can move towards the state in a very robust way. So a way to think about it, that's what I call minimum frustration. A way to think about it comes here. I could imagine this quantity Q, we'll be talking a lot about it, but all you have to think about it now is that quantity Q, yes. Yes. Okay.
When you have a protein that changes conformation when it's binded to, I don't know, some other molecule, the graph of entropy and energy, like you have two values?
Yes.
Yes or no?
Yes, so let's think, this is a very good question, and I'm going to be very cavalier on my answer, but you're going to see that's going to be a good answer for what you're trying to do. What you have here is, what I'm telling you is the simplest possible problem I can describe you. How a single protein lives in life. Okay? If I go to what you're telling you, that I have two proteins. Now you have to think about it. Why this particular system, if I think these two proteins are separate, why they are happier when they come together? Okay? And again, that's the next stage of sophistication to these problems that tells you that now, if the two systems are apart and these proteins are separate, you have a higher entropy state than the bound state. So how do you have this binding energy creating additional stability to my system? So that now I prefer on the pair. So I would have to reframe this entire problem in the same way to do that. But to give you an example, there are cases that you observe. For example, you look at these proteins, and I told you the concept of minimal frustration we describe here that tells you that locally, you're trying to make every state locally doing. You can find some proteins that fold, but they have some local areas that are unhappy. And that's normally an indication that that protein has a binding partner. When that partner comes, then it makes that interface happy. So this concept can help you as you move forward. There are many proteins, the simple ones, they are happy all the way around. Right? There are proteins what people call disordered proteins or intrinsic disordered proteins these days, where these proteins are disordered. They never can get order unless they bind to something else. Right? They themselves don't have enough bias. They only get enough bias when the two things coming together. So you could imagine if I cut protein in half, the two halves don't fold because they require long-range contact. So there are lots of stuff where this concept gets generalized. But these are very good questions. Start to think about it. There are many other cases. Proteins have multifunctional states. How do I deal with that? There are many other things you're going to bring to this discussion that you can bring to them. That's a very good one to come here. So in the concept of a single protein here, let's just use that to finish the lecture. I know I'm getting out of time. I have this Q. And this Q tells me that how ordered you are. So you go all the way from native to the states that are completely unfolded, they're completely disordered. And you're going to have many states, like I show you on the case of the two-dimensional, only had 3 states on that case. But in general, in real proteins, you have many different levels of order. Okay? And what this problem is telling you, a good unfrustrated system tells you the more similar you are to the native structure, your energy is also lower. So you want to have sequences where improving order and minimizing energy are equally good. If you have a protein folding, I don't care if the right start to fold from the right or start from the left, from the middle, means anything that's become more native becomes more attractive. And the system tells you the more you become attractive, you come down. If I imagine I have no hysteric effect that you have, because you cannot have crossing chains, there are many other problems that come to that. You expect this protein, as you're coming down in states more order, you can come down, makes no difference what you have. And you start just to bring yourself down in energy. Like I said, because the geometry, that's not ideal. But that's what the funnel idea came into protein folding. You're trying to funnel towards the native state. So the more funnel-like you are, the more folding-like you are. But that requires this idea that interactions that exist on the native state tend to be attractive. And interactions that exist on the native state tend to be repulsive. That's what makes protein folding very complicated. Because not only you're trying to get this state attractive, you're trying to make the states you're competing against, repulsive. Right? And that's the concept that you're trying to get. Right? So the idea is, if I go back to this slide here, trying to make sure this state dominate over how the trap states leave, and that's what I call TF over TG. That's the folding temperature, tells you how stable that state. TG is the stability of the states you're competing against. And designing is a very complicated problem because of that. Sometimes if you try to overstabilize that state, you may also create some trapping states and you don't notice on your, so I can tell you that optimized ratios is not an easy thing in terms of simulations, okay? So I think we're over time. I want to stop here. You got the feeling of what you're after. And we start, but I want to show you here that this idea is not different just the other problem. The other problem, we had 3 states, right? In a single 6-chain, but that's the concept, how as we evolve and form more contexts. And what I want is to have a situation where you are either stable here or you are stable in a native state. These intermediate states, actual thermodynamic, they exist with very low probability. Okay? Thank you, guys. I think you have been sitting for long enough. Yes?
So tomorrow will be another extended discussion on these topics and going on to other systems. And then the following 2 days is going to be practical things that we'll be doing. So did everybody get the message about the software that you need? Yeah. Did anybody have any trouble with any of it? Installing it? I installed it, but I don't know if it's working. Okay. For the virtual box, there was a link for the image. Yes. So you should just be able to say.
Come to the front. Come to the front.
And if you hit import and you just load that image, it should show up in the menu. You just double click it, and if it opens up and looks like a computer, it works. It's a virtual box for a Linux machine. So if that works, then for Thursday, everything is already set. It's all precompiled and prearranged on that image. On Wednesday, we're just going to look at some molecules, and for that you use VMD. If you can open VMD and it shows a little thing, VMD spinning around, VMD is working, as far as I know. So if those 2 things work, then we're great. If there's any problems, let me know. And today and tomorrow and even Wednesday, I can help you troubleshoot your machines. But they should be pretty robust how they're set up. So just let me know, okay? I'm happy to tell you how to set things up on a Linux machine if you have a Linux machine. That's not a problem. That's actually what we always do, but I didn't expect many people to have Linux machines.
Maybe you want to set a time where they can look for you if they have a problem.
Yeah, I don't know what your schedules are. Do you have things all day long, or is there a big gap somewhere in the day? Because my schedule's open.
So this afternoon they don't have classes. You are not having anything to do. On tomorrow, Wednesday and Thursday, they will have seminars. It'll be a special week with seminars.
But today, let's start today, because they're going to start with simulations tomorrow, so it's important if someone has trouble to look for a poll today to make sure you guys are up to speed for tomorrow, right?
Okay. So I think they can meet you on the afternoon downstairs in the first floor.
Whatever time. Just tell me the time and I'll be there and I can help.
Yeah, I think during...
Choose a time. The first one to choose the time is going to be the time. 2 o'clock? 2 o'clock on where the tables are.
Yeah, in the first
Go
floor.
down the table on the first floor.
Okay?
Hold me there. Bring your
Okay.
computer. Let's make sure everything's working so you go to the desk because of the time tomorrow.
Perfect.
Great. Thank you.
The coffee break will be upstairs, guys, in the first set of tables, you know, like the long ones. Okay? Perfect. I came to give the message.